thank you to Dr. Lin for the introduction and for the invitation, and allow me to have this opportunity to share some of my research works. So I see this is a very good platform to connect researchers with a similar background. Um, and The title of my talk today is the inference and the analysis of the external communication signals using exFINDER. So first, we know the process of the development of the multicellular organisms starting from one single cell. So it starts from one single cell to multicellular tissue to organ and all the way to organisms. And during this long process, we have different very important biological processes like cell division and colony formation, cellular dynamics driven by gene regulatory networks. And I think the thing that we are interested in here is the interaction between the cells. So that's the cell-cell communication. So the cells interact through communication to coordinate biological processes. So a cell can send, receive, process, and transmit signals with its environment and with itself. So cell-cell communication is the important way to coordinate biological processes such as cell differentiation, immune responses, and tissue repair. So this is a schematic showing the different steps in cell-cell communication. So first we have a ligand, or we call that as a signal. And so the ligand can be a lot of things. It can be very small molecules, or can be the protein, or can be a hormone, even can be a metabolite. So right now in this research, we only focus on the protein signal. And if the right signal, if the right ligand find the corresponding receptor, so the ligand can bind to the receptor. And then once the ligand binds to the receptor, the receptor is activated. And then the receptor can activate its downstream genes. Usually they are transcription factors, or we call that TF in short. And then once the TF is activated, it can regulate the target gene expression. And then so we can separate this into the following three steps. That's a ligand receptor binding, receptor TF interaction, and then TF activates the target gene expression. So these are the structure for the cell-cell communication we are considering in this research. Of course, we can. So it does not have to be, or in a reality, maybe much more complicated than these three steps. It can have a lot of genes involved. But we separate this into these three basic layers. And then what we usually do nowadays is that we took the tissue, we took a sample, and then we do the sequencing. Then we have the single cell transcriptomics data. Then the data is basically a big matrix, right? And each column representing an individual cell, and then each row representing a gene expression in different cells. So using the single cell data, we can infer the cell-cell communication. So this is a picture generated using cell chat. It's also developed in our lab. And so this shows the communication between clusters. So each dot representing a cell group. And then we can, using the single cell data, we can infer the communication between different cell types. So there are a few examples of the recent studies. So first, the epithelial mesenchymal transition, or we call that EMT in short, it's very important. During the EMT transition, the epithelial cells lose the cell polarity and adhesion. Then it will gain the migratory and invasive properties to become mesenchymal. So this is very critical in cancer metastasis. And so to study this, there's a paper. In this paper, they use different inducers to promote these EMT transitions. And they added three inducers in their experiment: the TGF-beta1, EGF, and the TNF. Since these three are manually added, so they're not necessarily highly expressed in the cells that they measured. So the TGF-beta1 has a very low expression in the single cell data in those cells that they collected. And the EGF and the TNF, these two inducers are not even captured in the data. So they are not included in that single cell expression matrix. But they are still very important. They can clearly, you can see from the figure that's from their paper, they can activate the EMT process. And there's another paper working on the zebrafish neural cluster cells differentiation. So the neural cluster cell is very important. It can differentiate into different cell types with different functions, like pigment cells, skeletal cells, or neural and glial cells. And then this paper studied the neural cluster cell lineage in zebrafish. And the left-hand side, this figure shows the single cell data they collected. So basically, this is a UMAP that we are very familiar with. So these are the after the annotation, then they identify those green cells are neural cluster cells, and then they want to study the differentiation trajectory of these neural cluster cells. And then all other colors, these are non-neural cluster cells. So they think they are more focusing on the neural cluster cells, so they remove those non-neural cluster cells. So they only took those green cells, and then they did further analysis. And then in their analysis, they identified different clusters that may differentiate from the early neural cluster cells. So the lineage is shown in the bottom as the early neural cluster cells, then move to temporary transition cells, and then later differentiate into cells. But all these analyses are just using the green neural cluster cells, and all other cells are just ignored in their paper. So why I want to mention these two research works, the reason is that the limitations of the existing computational method on cell-cell communication inference. So for example, like I said, we have a measured cells, and then based on these measured cells, we have the single cell data. And all these, so current method only focus on analyzing the cell-cell communication within those measured cells. So we have lots of very good, very powerful computational methods like cell chat, neural chat, NIDDK, iCellNet, and they all have their strengths, and some may have very high resolution, even to single cell resolution cell communication, and then some method can combine the spatial information to infer the communication. But all these methods only deal with the signals sent by the measured cells, so they only can infer what they have in the data. But in reality, it's highly possible that those cells are receiving signals from outside. We call outside is that, for example, the unmeasured cells can also send signal to the measured cells, because the measured cells come from the sample, right? The sample is a small piece of tissue that we took from the animal. And then, of course, the cells that are surrounding those samples can still send signal to the measured cells, but those cells are not captured in the experiments. And also, like the EMT paper, they manually added the inducers to the measured cells, but those inducers are not expressed in these cells, so it's not captured in the data. But these inducers can be very important. It can drive the EMT process. So we want to have a method that can identify those, we call those external signals, that can have a very significant impact to the measured cells. So that's the basic logic. And then so the logic behind it, how can we infer what we do not have in the data? The logic is, so first we need to have those signaling networks with these four layers from ligand to receptor to TF to targets. And then once we have this network, the basic logic is that, okay, we found this target is activated. It has a very high expression level. And then we look from the bottom to the top. Then, okay, these upstream genes, the TFs are also activated. It's very high expression level. And then the receptors also activated, but the ligand is missing. Either the ligand is not captured in the data, or in the data, it shows that the ligand has a very low expression level. But all these genes are activated, so they must have received something to activate this pathway. So there must be an external signal coming from outside. So that's the basic logic. And then so to have this computational method, there are a few challenges. The first one is how to infer the network, right? We have to have this network first, and then use the network and the data to identify the external signals. And then, of course, we may have multiple external signals, how to identify critical signals. And then if we have multiple signals and multiple targets, how to also identify the critical receivers or the critical targets that receive the most signals. And this is just one network. What if we have multiple signal networks? How to compare the overall signaling strength between different external signal related networks. And also, once we have the network, we want to know how to infer the biological processes that are associated to these networks. So these are a few challenges and a few goals that we want to achieve when we develop this method. So this method we call that exFINDER. So it's the first method to identify the external signals. So it's published on last year, and it's written in R. It's R package. So the basic is that the overview of the workflow of this method, we have the input is that the method of course, require the single cell transcriptomics data. We require the expression matrix, prefer the scaled and the normalized matrix. And then we have our own exFINDER database. So the exFINDER database, including those three steps, right? Ligand, receptor to TF, and the TF to targets. So we did not build this network from the beginning. We integrate several public available datasets, and then we made this available for human, mouse, and zebrafish. So this is the exFINDER database. And another one that's required to use the exFINDER is the user selected target genes. So unlike some of other methods that can infer signal network, or usually from top to the bottom, that's we starting from the ligand and then infer the receptor TF targets. Here we build the network from the bottom to the top. The exFINDER asks for the user selected target genes. Let's say, oh, I see these genes are activated, but I want to know why. So these genes are the target genes, and then starting from the bottom, you can build a network. Of course, so how to select those target genes can be tricky. So there are optional analysis that can be used to help selecting the target genes. So first, can be already become a routines discovering the marker genes. If you're interested in the activation of one particular cell type, you can use the corresponding target genes as the corresponding marker genes as the target genes. And also you can do trajectory analysis, and then if you're interested in a specific group of cells along the trajectory, you can use the high expressed genes of that group as a target genes. And another thing is that it's another paper that can infer critical transition signals based on different cell groups. And then you can use this, I think the method is called biotip. You can use this method to identify critical transition signals and then use that as target genes. And after we have those inputs set up, then so first is that we infer the network from the bottom to the top. We have the targets, and then we have the database. Then we found their corresponding TFs that can activate those targets. Then we found the receptors, and then we found the ligands. So this step is pure only using the database. Is not we are not using the single cell data. We just do have this structure. So this is basically a graph. And then once we have this network, we can use the single cell data. For each gene is each dot. So each gene in one cell type will have, of course, we can calculate the average expression level. And then based on the average expression level, or we can refine this network using the single cell data. For example, we want to find what are the targets that are activated, and we want to find that all these downstream genes are highly expressed. And then we want to check among those ligands, maybe some of them are also highly expressed, and some of them are have a very low expression. For example, just like this figure shows. So the ligand, these two may have high expression, and the rest may have low expression. And then, so the ligand with very low expression and the downstream are activated, then these are potential external signals. Then using this, we can construct this directed graph. So each arrow shows a signaling direction, and then representing these two genes are interacting with each other, and the size of the dot representing the average expression level for this gene in this cell type, and then based on this graph, we can predict the signaling strength. So the mathematical model here is relatively simple. We just, based on the mass accuracy law, we use this Hill function form to predict the interaction strengths between two genes using the gene expression levels. Then so based on the signaling strengths, we can color those edges to representing this strength. Then this become a weighted and directed graph. So each edge is weighted by this strength. And then once we have this this graph, we can do more quantitative analysis. For example, if we have multiple ligands, we can calculate the signal outflow for each of them, and then we can rank them, and we can find, oh, maybe this one had the most signal outflow. And then for the targets, we can calculate, then we can compute. So maybe, let's say this target has the most signal inflow. So that indicates that this is the critical ligands, critical external signals. This is the critical targets. And once we have multiple these weighted and directed graph, we can do classification for these networks. Then we can see, okay, maybe some of these networks have a similar structure, and then they will show in one cluster, in the same cluster, and then we can zoom in, we can further analysis, we can take a look to see if they are related to similar biological processes. So these are the first part of the quantitative analysis, and then to compare for one single network, it can be pretty big. It can be maybe have over 100 genes involved in this network, but sometimes we may interested in a subnetwork. We may interested in some particular genes. Then we introduce this activation index. So this index can quantify the activation level for each network. And then you can compare this between different networks, or you can compare the activation level between a subnetwork and the full network. For example, if we have a very big network and then we extract a subnetwork, and then we can compare the activation index, and then we can see, okay, if these subnetworks are critical play a critical role in this whole network. So this is one way to do that. And also there's another function is that if you have roundabout tip, if you identify some critical transition signals, then exFINDER can help identify the critical transition signal related signaling networks. So in these networks, the CTS, the critical transition signal, are these two TFs. Then we can figure out why these two are so important in the transition, and what genes will they activate. And the last part is we can based on the network to do goal-term analysis, and then based on the goal-term analysis, for each goal term, we can grab all the genes that related to this goal term, but the networks that we use may not cover all these genes. So we can compare, we can quantify based on one network, we have multiple goal terms, and which terms that have the closest relationship to this network. So we can rank them based on two different methods. So these are the basic functions, the main functions for exFINDER. And then for benchmark, we compare exFINDER with CellChat and iCellNet. So the first step is that exFINDER can recover ligand receptor pairs inferred by these two methods. So we want to see that if they can do that, if they can infer those ligand receptor pairs, can exFINDER also do that? We use this human skin dataset that have these 4 cell groups. So this dataset is also an example dataset in CellChat. And for target genes, we use top 10 marker genes for target genes for each cell group. And then the result shows that for most of the ligand receptor pairs inferred by these two methods, exFINDER can capture over 80% of them. So there are maybe 1 or 2 cases that is around 50%. We think one of the reason may be because that we know CellChat use a different method to calculate the average expression level. And then that's probably the reason why that we identified slightly different highly expressed ligand and highly expressed receptors. And also another reason is that those two methods purely focus on ligand and receptors, but exFINDER is starting from the downstream target genes all the way up to infer the ligand receptors. So these can result in different inference results. But overall is that exFINDER can capture most of the ligand receptor pairs that other methods do. And another thing is that exFINDER can identify the ligand that is lowly expressing in the measured cells. So this is example. We use the FBN1 plus fibroblast cells, the markers, as a target, and then we infer from the bottom to the top. We find those ligands, and then we check these ligands expression in those 4 cell groups. And then the principle is that the ligands inferred by exFINDER does not have to be highly expressed, because some of them like I said, it's maybe external signals, but their downstream genes must be. So in cell chat and iCellNet, if these APOE plus fibroblast cells, if these cells are not measured, like if these cells does not exist in the dataset, then this APOE will not be identified as a communication signal. For example, if we do not have this data, do not have those cells, then in their method, APOE does not have a high expression, so it's not significant. So they will not be considered this as a communication signal. But exFINDER can still capture that. exFINDER thinks that. So what exFINDER did is that exFINDER thinks that okay, the marker genes for FBN1 plus fibroblast cells are activated, and all the upstream genes are activated. So yeah, it should be some external signals. And still, even if APOE is lowly expressed in those three cell types, even if we did not have this, but this can still potentially be an external signal. And then if we can clearly see that, of course, if we include these cells, we can identify, okay, yeah, it's highly expressed in the APOE plus fibroblast cells. So this part, so the first part is that just to show that what they did, we can do reasonably well. And the second part is the unique part of the exFINDER. That's what they are not able to do that. Um, and so the next part is the couple applications. Come back to the EMT paper. So exFINDER can identify all those external inducers in different cases. They use three different datasets, and each dataset they use these three inducers. And in all cases, exFINDER can identify those external signals. So even if they are not captured in the data. So in the data, we do not have this gene, but exFINDER thinks that, oh, it still can be a potential external signal. And then for the zebrafish paper, what they do, they infer these neural cluster cell lineage and only using the neural cluster cells. And what we think that we want to see, for example, we took the skeletal cell as an example. We want to see if these cells received any external signals. So we took the marker genes for the skeletal cells, and then to run exFINDER analysis, we find this ligand. This ligand might be the external signal. And then by check the data, we found that this ligand has almost zero expression in the neural cluster cells. But in the non-neural cluster cells, at least in epithelial cells, they have a relatively high expression level. So it's not that obvious compared to those receptors. But if you can only check this gene, you can see that it has a very high expression in the epithelium cells, but it has almost zero expression in those neural cluster cells. So this suggests that in the future, if we in experiment, if we collect a lot of cells, some of the cells seems to be unrelated or seems to be unimportant to the research, but they can still send signals to those the cells that we are interested in. So this suggested that in this case, those non-neural cluster cells serve as external unmeasured cells because it's not included in the original paper, and it can still send signal to the neural cluster cells and potentially activate those marker genes for the skeletal cells. And then we want to go one step further. We want to see what if we by the way, this is the analysis directly from cell chat, suggesting that if we including all those cell types, cell chat can also identify the communication between epithelial to skeletal, and it's pretty strong. And so these results also support our finding. And then we want to go one step further. We want to see what if there's an external signal outside all those cells, right, including non-neural cluster and neural cluster. Then we find this ligand. So it's not expressed in the data, but we can still infer. So it's possible to activate the receptor RPS19 and then to activate those marker genes for the skeletal cells. So that's something new. To verify this may need additional experiments. And then we have the CTS related signal networks. So these two are the CTS critical transition signals that identified by biotip. Then in exFINDER analysis, we infer those signal networks. And then we want to see why these two are critical transition signals. And it turned out to be that these two can activate both pigment markers and skeletal markers at the same time. So these two can play an important role in the transition from early neural cluster cells to the pigment and the skeletal cells. And the last application is, so this paper using single cell data to identify early diversity of the sensory neurons forming in the mouse. And then, so this is the UMAP with trajectory analysis results. So they infer those two branches A on the top and branches B on the bottom. And the first step is that they think the latent neural cluster cells, these red cells, are the starting point for branch A. And then what we found is that, so this is schematics of this trajectory. So using biotip, we found that, okay, the latent neural cluster cells can be critical in the transition. But we want to know why. We want to know what signals can activate those latent neural cluster markers. So we use the markers for these latent neural cluster cells, and then we infer what can activate those markers. So these are a starting point for the branches. So we want to know even earlier, right, what can activate those. So we find these three signals that can activate these two receptors, and then sending signal activate the marker genes for the late neural cluster cells. So these results are supported by previous studies. And then we can, among those three signals and those targets, we can identify which this one has the most signal outflow, and this target has the most signal inflow. So we can identify the critical external signals and critical targets. And then next is that we see in our original research, the branch A and branch B has the kind of intersection. And we want to know, so this group are unidentified in the paper. They call that unassigned number 1. So this group are kind of at these intersections. We want to see if this group can also contribute to branch B. So there are early divergent genes provided by the paper. We use these genes, the early divergent genes of branch B, as our target genes. So we want to test this hypothesis that maybe some external signals can activate both the receptors in both these two groups, and then those receptors can activate the early divergent genes in branch B. So that means that these two can also contribute to the differentiation along the branch B. Yeah, and then we infer there are lot of possible external signals, and then two receptors that are related to activating those genes, those early divergent genes. And then some of the results are supported by previous findings. And then based on focus on these two receptors, we found that these two receptors have comparable expression in these two clusters. So that means that based on these external signals, they can activate these two receptors at the same time, and then these two may contribute to the branch B. And then using this network, we can perform the go-term analysis and then compare this function in exFINDER to compare the to rank the top 5 biological processes that are related to this network. So this is the third application that shows that exFINDER can potentially help us to compare and identify the important trajectories. And then so the overall the summary of this method is maybe the first method to identify these external signals, and then you can also infer the signaling networks, including those downstreams, and then do quantitative analysis. Central to this method is signaling paths that link the external signals and the target genes. That's the network, and the edge weight. So number 1 and number 2 combine these two. We use these two to construct those weighted and directed graph. And then it's our package available online. So basically we can go back to the 3 questions. What external signals influence cellular behavior within a single cell data? So exFINDER can identify those external signals and then identify the critical external signals and target genes, and also link the signaling network to important biological processes. So these are how exFINDER can answer these 3 questions. Of course, this is so since this may be the first method to do that, also it has a lot of potential improvements that we can do on this method. So first is that, so I list a couple. So first is that right now exFINDER cannot do comparative analysis at the same time, like parallel analysis on the 2 conditions. If we want to have a comparative results, we can do exFINDER analysis twice using different data on the different conditions. But it will be good if we can have a comparative analysis function between conditions, and it can help review the critical signaling pathways regulated by disease or perturbations. And also, if you can utilizing multimodal data, right now we only use a single cell transcriptomics data. If you can use multimodal data to refine the inference and predictions, we will have more accurate results. And also, since we are more and more there right now is more and more spatial transcriptomics data. So if we can integrate the spatial information, and maybe in the future, if we have multiple time points, so that means if we can integrate the spatial temporal information from the data, then there'll be very helpful to identify cell-cell communication, because we can consider the time is one cell appears at 20th hour cannot talk to a cell that appears at the 50th hour, right? And then 2 cells which are further away from each other cannot, or cannot, just it's very unlikely that they can communicate with each other. And also we can do database refinement. Right now the database, the good thing is that they contain lots of interactions, but some of them I check the sources. Some of them may based on text mining, but that's not very reliable. And some of the TF may not be actual transcription factors. So if we can do a database refinement, that will be also very helpful. And also, right now we do not have any analysis to quantify the pathways of significance or robustness, like p-value or bootstrap tests. So if we can include this part, it will have more information to rank those signaling pathways to identify which of these pathways are very important. So based on these possible improvements, so there's one ongoing project I'm working on. So this project is using so it's called cell signaling networks discovery from multimodal data. So determining activated pathways only use single cell data is not enough. You only use single cell RNA-seq data is not enough, which only capture the mRNA levels. So the multimodal data offers greater evidence for accurately inferring activated pathways. However, no existing method can systematically infer and evaluate signaling networks using multimodal data. So we want to have a new method that can infer the networks and processing the multimodal data and use the multimodal data to evaluate those pathways. And we want to do comparative analysis on the different conditions. So this project is collaborating with biotech company called Cell Signaling Technology. And so the basic concept is that we still have the ligand receptor binding and TF activation, target gene activation. But right now, for the RNA-seq data, we are at this level. What if we have proteomics data? And then what if we have phosphorylation data? And then related to phosphorylation, what if we have kinase information? Then if we have all this information, if we can include all of them, then for one pathway, if they are activated in all those levels, then we are very confident and comfortable to say that, oh, we see this pathway is truly activated and functioning. So this is the concept. And so the CST, they can provide proteomics data, phosphorylation data, and they have their own database to infer the kinase information. Then, so the basic overflow of this method is that we have input genes, we have sender genes, we have receiver genes. These genes are user input, like what genes you are interested in. Usually we prefer marker genes. Of course, you can throw all the genes in the data, but it will give a hard time to your computer. And then we have single cell data, proteomics phosphorylation, and the kinase library, kinase information for example, for each gene, for this gene at this phosphorylation site, what is the kinase that can phosphorylate this substrate. So based on this information, you can process all this data and then infer the pathway, perform quantitative analysis, and then based on the quantitative analysis, we can do evaluation for all these pathways to see which pathways are highly upregulated in one condition, which pathways are greatly downregulated in one condition. And also for different pathways or different networks, we can do classification via graph learning. So the good thing for this one, the powerful thing for this method is that usually we have pathway inferred without using multimodal data. Maybe they can infer a lot of pathways. And then without using multimodal data, some of the pathways are ignored because they are not highly expressed, or if they are not differentially expressed, for some reason, they are ignored. And using multimodal data, the good thing is that for those pathways that highly expressed in single cell RNA-seq data, but not active in the proteomics level or phosphorylation level, then this we call that false positive, right? And then this new method can identify these, so we can ignore those because they prove to be unactivated. And then for some of the ignored pathways, maybe they are not differentially expressed in the RNA-seq data, but they have very high proteomics level and phosphorylation level, then we can impute them. So we can save them from being ignored. So these parts. And then since we have more data, we have proteomics data, we have phosphorylation data, we have a wider range for selecting those sender genes. For single cell data, we can only use the single cell data to find maybe marker genes or highly expressed genes. But right now we have more data. We can including more genes. Like the genes have a high proteomics expression level, so we may identify new pathways. So these are some advantages for this method. And to the end of my presentation today, so I want to thank to my postdoc advisor, Dr. Jing Ye, and my collaborator, Dr. Tom Schelling, by providing the zebrafish data, and Dr. Daya is from the senior scientist from the CST. We are working on this multimodal data project, and the agencies, the NIH, NSF, Siemens Foundation, multi-scale self-aid research for supporting my research. And that's all for my presentation today. Thank you.
Thank you, Changhan, for your fantastic presentation. So now is our Q&A session. You're welcome to unmute yourself if you have any questions. And there is some question from the chat during your presentation. So first question is from Dr. Ronald Markers. So very nice project. It sounds in concept similar to the NIH Common Fund project. Links library of integrated network-based cellular signatures. Links has transitioned from the NIH Common Fund but maintain database on cell types perturbation cellular responses. Have you looked into how exFINDER might intersect with links data?
Oh, I just saw the question on the chat. See library of integrated network based services. I think that's a very good point. Unfortunately, I'm not very familiar with links. I wonder if you could explain a little bit more about this. It looks very interesting.
Yes, this is Ron Margolis. Links was a project supported by the NIH Common Fund, and what they tried to do was create a three-way grid between cells, perturbagens, that is, could be ligands, or could be environmental compounds, and so on, and then cellular responses. And they were looking for connections between pathways and creating nodes and intersections between those pathways to predict cellular behavior. The project has transitioned from the NIH Common Fund, but still maintains this database with extensive data. And it's linksproject.org. It might be worth looking into it and see what they have, because it sounds very similar to some of the things that you're trying to do.
Yes, I think, first, thank you for the information. I think the basic, I can briefly see the basic mathematical concept is like still graph based, and then so basically we infer these networks building the graphs, and then they may have additional layers or additional rows in these networks. But yes, of course, I think exFINDER can, of course, have some common functions that can be used for this project, or even not, we can maybe add one or two several modules that can be applied to this project. I think so. But of course I'm more than happy to look into this project. Thank you.
Okay, thank you. So our next question is from Dr. Ashley Shah. So can your algorithm be used to analyze whole tissue RNA-seq data?
So I think the whole tissue data, what I can see right now is the whole tissue data is that we have a lot of cells, and as long as it's still single cell data, exFINDER can of course can do that. It's just like it may be challenging for maybe we need to use HPC to run it. And I think the next one is very interesting for cross-organ communication based on multi-omics data. So first, right now, I think the concept of exFINDER is all possible for used for cross-organ communication. But right now, I can see there are two ways to do that. The one is that we can build another cross-organ communication database. but I don't think that's available right now. If that's available right now, we can just switch the database and then we can use exFINDER. But another one is that we can, so if we have two species which potentially activate one target from one species, then we can compare for set one as a external signal and set another one as an external signal. The additional steps that we need to have is that we need to compare the contribution from these two species. If I think it's a similar concept. For example, we have human genes activated, we have human signal, mouse signal. If the human signal is very weak, have a very low contribution, but the mouse signal will have potential very high contribution, then we can infer this across, not cross species. I'm sorry, I thought it's cross species. It's a cross organ communication. Oh, I see. Sorry, because so the cross organ communication, yeah, it's similar. So one organ have the signal, and another organ, and the organ that activates those target does not have the signal. Then we can infer the communication from one organ to another. Yes, that's the similar concept. I can see right now there are few things to do. The first one is that we need a database for those long range interaction signals. And another thing is that we need to consider the signal diffusion cross organs. I think we probably need to build another math model to describe that. And also for tissue data, I can see including more cells. So it's better to consider machine learning framework on this one. Yeah, I think it sounds very interesting. Thank you.
Thank you. Okay, and there is another question in the chat. So I want to share this exFINDER resource with my student in the class. Is there video or tutorial available for me to use? And then are there any required skill for them?
I think so the video resources, one is, of course, today's presentation. I think another one is that we have I can type that gene pals. I think it's this one. So this is a club that at UCI I was invited to give a tutorial video. I think it's available on YouTube. And of course there's tutorial documents online. And then for required skills, basic R programming and familiar with single cell data and Seurat. Yeah, and if they are familiar with cell chat, that's a plus.
Okay, thank you. Are there any more questions from the audience? If you have any more questions, please feel free to send us an email, or I will forward the question to Dr. Changhan He.
I think Dr. Xia has a.
Okay, yeah, Dr. Xia.
Hi, thank you. Really beautiful presentation. Yeah, I'm the one have the question in terms of whether your algorithm can use cross-organ and also, the whole tissue RNA data. The reason I'm asking that is because I'm work with the NIH Common Fund called Molecular Transducer Physical Activity. We have a large data set that basically animal we trained with exercise, and we have 19 tissues of RNA-seq data, and also we have multiple tissues, the multi-omic, basically proteomics, lipidomics, metabolomics data available. Those different omics are actually all used from the same tissue source. So it's really a good sample data set for you to look into whether your algorithm can use to analyze cross-organ. Because I think another way to think about the cross-organ is whether there's a secretory protein, right? In a different, whether they're upregulated. That's one thing. And the other thing is whether there's, receptor of that secretive protein expressed in other tissue, whether they are overexpressed, by exercise. I think that there's lots of ways you may look into this. And the good thing about it is we have RO3 funding mechanism out now. It will build, the deadline is December 27th. So if you're interested, you'll still have time. If other people interested, you also, you can look into, I'll put the announcement on the chat so everybody can check on it.
That's great. I think, yeah, it's very interesting. You already have so much data, and I think the crossover communication has a very broad application for disease like diabetes, like the disease that the NIDDK focus. Some diseases are involved in multiple organs at the same time. And yeah, I definitely need to willing to learn more about this project, and maybe we can talk about like what are the potential signals that can travel across
Yeah,
organs.
if you really want to, the keywords to search the portal, it's MotorPack Data Hub. So if you do a Google search, you'll find the sites where you can find how the experiment was done, what's the data set, you can download data set, it's all there.
Oh, great. Yeah, thank you.